Okay, good morning all. It's a pleasure to be here to give a talk to you guys. It's very rare that we get the opportunity actually to talk outside of the scientific arena, so it's a pleasure to come over. It's my first time over at the village, and I've got a few of my colleagues here from my lab have come across as well, so feel free to have a chat and hassle them later on if you like, and thanks to Nicole and others for organising this. So, as Nicole mentioned, I'm going to give you a bit of an overview today of what sort of research we do in lymphoma over the Diamantina Institute, which is across the road here at the Translational Research Institute on the PA Hospital campus. So it was a quick 10-minute walk for us this morning. So I'll start off giving you a very brief overview of who I am, where I've come from. So I studied in Brisbane. I studied at the University of Queensland in biomedical science, and then I did my PhD at UQ and also at the Queensland Institute of Medical Research at Hurston, back then with Dr. Andrew Nicol, and this was in haematology, and we're looking at ways to use standard treatments such as chemotherapy to induce an immune response against cancers and try and optimise ways that we could do that, with the hope that we could then try and drive more of an immune response against cancers we were targeting. After I graduated, I did my postdoc, so this is a term we call further training basically after we've graduated. Did 2 postdocs. The first one, which is at the Diamantina Institute, that was with Professor Ian Fraser, who a lot of you may have heard of. He's the gentleman who developed the cervical cancer vaccine. And back then we were looking at immune regulation in models of cervical cancer, so I sort of moved away from blood cancers and more solid tumour work. And then I spent 3 years down at the Peter MacCallum Cancer Centre in Melbourne, which is, I think, the only Australian hospital specifically designated to cancer. And I was with a very world-renowned tumour immunologist, Professor Mark Smith, down there. This is where we were really looking at developing immunotherapies for different types of cancer, actually, but this is where I really started work. Started working on lymphoma specifically, and we use animal models of lymphoma to test our therapies. And in 2012 I moved back to Brisbane and set up my group back at the Diamantina Institute, and soon after that we moved into the New Butte TRI building, which is a fantastic place to work. This is the team, and as I said, some of them are here today, so a lot of the work I showed today it's not me doing the work, it's these guys doing the hard work and the long hours in the lab. So these are the guys who we really have to appreciate doing the hard work. And I'll mention, I'll flick up some photos as we go along to attribute the work to the different guys in the lab. And obviously to thank these funding bodies, including Leukaemia foundations who have funded us in the past, because without these funds we can't do the research, and obviously it's very expensive to do medical research. Okay, so this is the TRI building I was mentioning just across the road. It's a fantastic place to work, fantastic environment. There's actually 4 different institutes within this building, so it allows a lot of collaboration between scientists, between clinicians, and being on site at the hospital, obviously we can engage in a lot of translational research and patient research. So come and visit us. We're always open to showing people around. Okay, so focusing on the Diamantina Institute, so it's an institute of the University of Queensland. We're split up basically into 3 major programs: cancer, immunology and genomics. And within these disciplines we study a range of diseases as listed there, and with a major focus on autoimmune diseases and cancer. You can see here blood cancers is one of our strengths. So where does my group sit in? We basically sit at the interface between the cancer and immunology programs because we're cancer immunologists. And as I said, we basically focus mostly on the blood cancers these days: leukaemia, lymphoma. All right, so what are our plans? What are our goals? What do we aim to achieve? Really to understand how blood cancers and their treatments, for example chemotherapy, monoclonal antibodies, affect the immune system, and then to use this knowledge to develop and test therapies with the aim to complement conventional therapies with our immunotherapies. And our motto, I guess, really is to improve treatment quality and outcomes for patients with advanced cancers through translation-directed immunological research. And the disease we're mostly focused on is non-Hodgkin's lymphoma, the subset of non-Hodgkin's lymphomas, B-cell lymphomas. Okay, so taking a step back, immunology is a very complex field, and I'll just touch on it briefly today. But the concept really in cancer is what we call this immune surveillance. So similar to how the immune system sort of patrols and surveys for viruses and bacteria and infections you may get, you may not know that it also does this for cancer, okay? And at least in the early stages, when you have a few cancer cells developing, the immune system can be actually quite effective at detecting these cells and eliminating them. So a lot of us may actually get cancer to some low level and be cleared by the immune system without even realising that we ever had it, okay? However, over time, if these, a lot of things happen, these cancers are very unstable. There's a lot of genetic changes in these cancers. And over time, the cancers are very clever. They're kind of like viruses, and they can manipulate the immune system. So what happens is we go from a potential elimination of the tumour to this equilibrium phase where there's a constant battle between the immune system and the cancer. Okay, and again, this might be undetectable tumour, a cancer at this stage. It might not be clinically apparent. There's this constant battle here, but we can't clear the cancer. And then worst case scenario is when the cancer gets to a point where it's able to overwhelm the immune system, okay? And we call this the escape phase. So they mutate, they change the mechanisms, they can evade, hide from the immune system, they can escape the immune mechanisms, and this is where we get clinically overt cancers developing, in a nutshell. Okay, so as immunologists, we basically we study this interactional fight between the immune system and the cancer. Okay, and it's really this balance between immune activation and immune suppression that we look at. And really, it's depending on what happens here, will largely determine the outcome of the patients and also response to some of the treatments. So, for example, in the cancer setting, there are a lot of things which really drive this immune suppression, okay? There's this dysfunction of the immune system. The cancer cells themselves can cause this. The treatments that patients are on are immunosuppressive a lot of the times. Certain environmental factors and also our genetics can all sway this immune suppressive phenotype, okay? And obviously that promotes cancer development and growth. What we try to do is intervene, okay, manipulate the immune system with immunotherapies, immunomodulators, which aren't themselves immune therapies, but they modulate the immune response, and also by considering, lifestyle factors to tip the balance towards immune activation. And we know if we can do this well enough, we can achieve cancer regression in certain types of cancers, and the hope, obviously, is for long-term remission. Because the good thing about the immune system, and I'll come back to this later, is once you've invoked a good immune response and invoke immunological memory, there's a potential for very long-term protection against the cancer, which a lot of other therapies can't achieve. Okay, so what is the immune system? I'm not going to touch much on this at all, except to say that there's a whole variety of different cell populations with different developmental pathways, and they all have very specialised functions in the body, okay, whether it's to clear a virus, clear a bacteria, etc., etc. Okay. We mostly focus on these what we call lymphocytes, because they're kind of the cells that we utilise to attack the cancers. But what's often not appreciated, at least in the blood cancers, is lymphoma as an example, is this is a cancer of the immune system, okay? The cancerous cells, the tumorous cells are in fact immune cells. So we have to take that into consideration when we're looking at interactions between the cancer and the immune system, and also looking at therapies, because often the cells we're trying to activate and promote for the immune response are also the tumour cells, which we don't want to be activating and promoting. Okay, so it's quite a complex environment that we're trying to intervene with. Okay, and the specialised cells that I'm talking about that can target the cancer, largely we're focusing on T cells and NK cells. Okay, two lymphocyte populations. And I thought I'd just throw this picture in here because I thought it was quite interesting, and quite often in science we don't get to visualise things too much at this sort of level. But this is a tumour cell here in blue, and this is a 4 T cells which are recognised and are targeting the tumour cell and killing it. Okay, so very nice image here. But this is an example of this sort of recognition and specific targeting of the cancer cells. Okay, I have to talk a little bit about T cell activation, so this makes more sense. My data makes more sense later on. So basically, we have a T cell here, and we have another immune cell here called antigen presenting cell. And basically, this cell's role is to instruct the T cells on how to behave, basically how to function. And depending on the signals a T cell gets will depend on whether it then can do its job or what type of job it does, okay? And there are three main signals that the T cell gets. First one really is interactions through the T cell receptor, which recognises antigen, okay? So these can be foreign antigens like virus particles, etc., which is foreign to the body. So that the T cells can recognise that when it's presented to them. But also in cancer, these APCs can present self-antigens, okay? Because the cancer is your own body's cells, okay? But they can be quite a little bit different. The antigens that come out from those cancers because they're mutated, etc., can be quite different. And occasionally the T cells can recognise that. However, in addition to that signal, they need what's called co-stimulation, okay? So as the name suggests, it's a secondary signal that they need to receive. This exposure here to the antigen is not enough. They need to get this co-stimulation to get properly activated. And people would argue there's also a third signal, really, which are these soluble factors that need to be signaled through the cell, which are largely called cytokines, okay, to get a full-blown response. So we focus mainly on developing vaccines, so different types of immunotherapies. Vaccines is one type. They're therapeutic vaccines, so we use these vaccines as a treatment. Why is this important? So I've mentioned that tumours can evade or escape immune surveillance, and that's often why these tumours develop in the first place. Patients are often immune suppressed as a result of their cancer or their treatment. And there's a large amount of evidence now to show that anti-tumour immunity can be generated, restored, or even boosted if we intervene in the right manner. Okay. Just bear in mind this is different from prophylactic vaccination. For example, the flu shot, where we're giving a vaccine to try and prevent us getting a disease. We're actually looking at a therapeutic vaccine, okay, so to treat existing disease. All right, so this has been our approach over the last few years. This is very wordy here, okay, but basically what we're doing is taking cells from patient, in this case the mice, so we work a lot in mice models. So we take cells from a mice, tumour cells from a mice. We load them up in a lab with this molecule which is known to activate another type of immune cell, natural killer T cells. Okay, so they're another type of T cell which have been shown to be important in generating immune responses against cancer. So this sticks to the surface of the tumour cell. Then we irradiate the tumour cells in the lab to stop them proliferating, because obviously we don't want to be putting back proliferating cells into the body. And then we infuse this as a vaccine product back to the patient. Okay, so basically we're taking the tumours out, feeding them this molecule, irradiating them, and putting them back in again. And the hypothesis really is that once we give this vaccine, these NKT cells specifically can recognise this molecule on the surface of the cells. That really activates them and also drives activation of other types of immune cells that we know are important in targeting the cancer. And the hope is that all these cells then work together and find and attack the growing tumour in the body. Okay, that's the concept. This is how we do it. It's very simple. So we give our mice tumour cells. EU-MYC tumours are basically a type of lymphoma, B-cell lymphoma. So we give the mice lymphoma. After we're confident that these mice have lymphoma, have disease, which we can detect in the blood by this technique called flow cytometry. We treat them with this vaccine product, and then we monitor these mice every week. We bleed them, or we image these mice with special cameras to monitor tumour development over time. Okay, and then we can determine how well our vaccine's working. Okay, so I'm not going to show too many data slides. This is a summary of a lot of work we did, which is now published. What you're looking at here is 5 mice. They've been anaesthesia. They've been put to sleep, put in under this camera which measures bioluminescent light. And the tumours, these lymphoma tumours, have this enzyme called luciferase. And when we give a substrate it lights up basically under the camera. So we can see where the tumours are in these mice, and we can track this over time. The top right here says 5 mice that haven't been treated with anything. This is 2 weeks, 2.5, nearly 3 weeks after we give the tumour. You can see here there's the tumour spread throughout the body. It's a very aggressive lymphoma that we work with here in the mice. However, when we give one single vaccine treatment, these mice at this time point are basically tumour-free. Okay, so at least in the early stages, that the vaccine is very effective at suppressing tumour growth. This is another way of looking at it. So this is flow cytometry, looking at tumour cells in the blood. So this population, the square here, are our tumour cells. These are in untreated mice. You can see, majority of the cells in the blood are tumour. In our vaccine-treated mice, very few cells in the blood. And basically, the outcome of this is a doubling in survival of the mice. Okay, so this is a survival curve. You'll see a few of these in this talk. As the mice die, you start off 100% survival. As they die, it goes down, and if they all die, the line comes down to the x-axis here. So you can see here, it basically an average of 20 days to sort of 35 days. So an increase in survival advantage basically with the vaccine. There's another way of looking at it. This is tumour growth in the blood over time. So this is time post-tumour inoculation and the number of cells in the blood. You can see here compared to the untreated mice which develop tumours very quickly. When we give the vaccine, there's this period here where the vaccine really suppresses the response, okay, and then the tumours grow out very rapidly, okay. So what we think is happening is that in this early period, the vaccine is generating a very strong, what we call innate immune response, okay. These are cells that can respond very rapidly to any stimulus, and this includes our NKT cells and NK cells. But then what happens is over time, that innate response, immune response, becomes weaker, okay, and probably eventually sort of disappears, and that's kind of the point where the tumour cells go, oh great, I'm not being attacked anymore, I grow out. So it was great that we're getting good suppression early, but obviously a problem here because the tumours grow out. So that perplexed us and brought us to the next part, and this is largely work done by Takumi, who's here today. We looked at ways of combining our vaccine that I just described with an antibody-based approach. So basically the idea is we use the vaccine to activate our cells, and we know from a lot of the work we did that the vaccine was very good at activating these, particularly these innate cells. This antibody here, anti-4-1BB, it's one of those co-stimulatory molecules that I explained earlier that T cells need to get proper activation, okay? And it's known from previous work that if you give this antibody, you can really promote the survival and persistence of these cells which are being activated, okay? And that's what we're aiming to do. So we generate good cells with our vaccine, but maybe they weren't being generated properly or they're dying off. We give this antibody with the hope that it allows those cells to persist longer and attack the tumour for longer, okay? So again, going back to this picture here that I showed earlier, the vaccine really drives this initial response here, this initial signal, and then this antibody which signals through as a co-stimulatory molecule really promotes the driving of the response and persistence of the response. And cutting a long story short, what the main finding from this combination therapy was that if you look at the number of these CD8 T cells, these T cells which attack the tumour, in this case in a lymph node, okay, so in an organ where we have the lymphoma, these are numbers of cells. So compared to no treatment or even vaccine treated, if you give this antibody, this 4-1BB antibody, you start getting expansion of these cells, and if you combine the vaccine and the antibody, you get this huge expansion of these CD8 T cells. So we thought, great, that's really what we're aiming for, because we know if we could generate good CD8 T cells that could persist, we've got a chance of controlling this tumour. Okay, again, this is the schedule, so very similar to before. The only difference now is after our vaccine, a day after we give the vaccine, we start treatments with this antibody. Okay, so it's every, I think, 3 or 4 days here for a couple of weeks. And then we monitor as usual. Okay, so this is basically the outcome of all this work, and you're looking at survival curves again here. And these are the 4 different treatment groups, okay? And I'll just direct you to the blue line here, which is our combination therapy. And you can see these are 2 different two different lymphoma cells that we use, okay, two different types of lymphoma. You can see here we get anywhere from 50% survival now, 100% survival of these mice long term with this combination therapy, which we weren't achieving with the vaccine alone or the antibody alone, which gives some protection compared to no treatment, but all the mice are dying here. So this was fantastic for us because it was the first time ever really that we could cure these mice from lymphoma and give long-term protection basically. So these mice, we've measured them out to at least 100 days here, and they still don't have any detectable tumour. We did another simple experiment then when we took these mice here, these long-term surviving mice, okay, that received the combination therapy, and we re-challenged them again with a tumour. Okay, so we do this to measure if there's any immunological memory. So these mice don't have any tumour, but they should have memory cells that were initiated by our treatment. So if we give them tumour again, hopefully they'll be able to, immune cells will be able to recognise a tumour and suppress that tumour growth. So the same tumour, we give the mice the same tumour again, and compared to the red line here, which is just naive mice and mice that haven't been exposed to anything before, which die very quickly, the mice that were re-challenged, again, there's a delay or improvement in survival, and again 50% of these mice also go on to have long-term suppression. This is with no further treatment, okay, so give them tumour cells and just wait for that immune response to do its job again. So it was obvious to us that there was some sort of immune memory that was generated with this combination therapy, which could control even re-exposure to tumour. And significance of all this really is that we're generating very strong and durable protection against the tumour by forming this immunological memory. We took this one step further in this model. So all that data I showed you was with a transplant model, meaning that we transplant tumour cells into a mouse. It's quite an artificial way of doing it. We also have these, they're called transgenic mice. So these mice develop lymphoma spontaneously over time. So I guess it's a more clinically relevant setting, okay? So these mice develop lymphoma in their own time at different timings. And the important thing is here that the lymphoma develops together with kind of the immune system. So there's all these interactions which occur that would naturally occur in a patient population as well. So we thought we'd test our therapy in this because we knew if the therapy could work in this model, it's a good therapy, at least in the mice. Okay, so we took 5 10-week-old mice, so these adult mice, and we basically did the same thing. We vaccinated, gave these therapies, did it twice, okay, so revaccinated again. This is work done by Brown and Doff. And again, we had the 4 treatment groups, and you can see here there's a lot more mice than I showed before, but the vaccine doesn't do a great deal in this setting, but surprisingly the antibody alone, the anti-4-1BB co-stimulatory molecule alone, was quite effective. It's proven to be quite effective. This is still not completed yet, this experiment, but it might be just as good as a combination therapy here. And we're trying to understand why that is, but in any case, we have therapies here which are quite effective against even spontaneously arising lymphomas. Okay, so to summarise all this, this is basically the mechanism of what's happening. So our vaccine that we give, the cellular vaccine, drives a very strong NK T cell response because they can recognise this vaccine. That then leads to a cascade of events which drives other immune cell populations such as NK cells and CD8 T cells to get activated. And we knew with this vaccine that we could combine, provide rationale to combine with this antibody, anti-4-1BB, which is upregulated on activated T cells. And what this basically meant was, rather than just getting this innate immune response against the cancer by these NKTs and NK cells, we drove a very strong CD8 T cell response, which we couldn't achieve with the vaccine alone, which can then participate in targeting of the cancer. Okay, so there's more participation there, and that's when we get good clearance of the tumour. Okay, so there's this combination of innate and this adaptive response, memory response, which was required to give good protection. Okay, I won't go into detail with any of these. This is just other examples of work we're doing. So we're looking at other ways to deliver this vaccine, and this is a PhD project for Ali in the lab. So she's looking at nanoparticles, so using nanotechnology to deliver this NKT cell molecule and tumour antigens as well to dendritic cells, which are one of those APC populations, to try and generate an immune response. And we're also trying to combine this NKT ligand to monoclonal antibodies which are available clinically, such as rituximab, which you may have heard of, an anti-CD20 therapy. I won't spend any time on that, but that's just to give you a feel for we're looking at different ways to deliver these immunotherapies. Okay, the other bit that a new focus for us really, which is still in its infancy, but this is work being done by Michael for his PhD project. We're interested in the concept of the crosstalk between the immune system and the nervous system, okay? And it's called cancer neuroimmunology. And specifically, we're looking at the effects of chronic stress and its impact on cancer development and also response to immunotherapies, okay? Which there's been a lot of anecdotal evidence showing that chronic stress leads to immune suppression, okay? And so that's why you may hear that, when you stress, you're more susceptible to getting colds and flus, etc. But surprisingly, not much has been studied in the area of cancer, okay? So we thought we'd tackle this. And I kind of see this as a vicious cycle. I call this a 6-cycle stress, immune, cancer cycle. So we're talking about chronic psychological stress, and as you can appreciate, in this day and age, a lot of people are under some sort of low-level chronic stress, okay? We know that somewhat this leads to immune suppression and dysfunction. As I've shown before, immune suppression can lead to development or enhance the development or susceptibility to cancer. And obviously, cancer is a very stressful disease, and when you've been diagnosed, it's more likely to lead to more stress, okay? And it perpetuates this cycle, okay? More stress leads to more immune dysfunction. Potentially, therefore, treatments may not work as effectively if you're immune suppressed. That's a theory, okay? And so we're using mice, again, mouse models to test some of this stuff. And very broadly, we use two main methods here, a physical method to induce stress in the mice and a drug pharmacological method. So the physical method basically is putting mice in these little chambers and individual chambers here, and we put this lever on top of them. What this does is prevents the mice moving around, okay? It doesn't squish them, it's a lever that sits on top of them and stops them moving around. And being a prey animal, mice don't like being confined to a small space, okay? That stresses them out. So that's one quite simple way of inducing stress physiologically. The other way is simply to inject mediators of the stress pathway. So the two main pathways are the adrenergic pathway, this is sort of the adrenaline fight or flight response, and the glucocorticoid pathway. And for example, cortisol, corticosteroid is a main mediator of that. So we can inject these molecules straight into the mice and induce a stress-like state in the mice. Okay. So just briefly, just a couple of graphs to show you that, again, these are mice that have been given lymphoma, okay? But prior to that, they'd be given this daily injection of isoprelin, which is an analogue of noradrenaline, so it stimulates this adrenergic stress pathway. And Michael basically maintains that treatment daily for 3 weeks. In the meantime, he gives them tumour. So basically, we want to see what effect this stress has on the development or the growth of the tumour. And you can see here, compared to, so these are mice getting a controlled treatment, the blue ones, mice that receive isoprelin, chronic exposure to isoprelin die much quicker, succumb to disease much quicker, indicating that triggering that stress pathway leads to faster development and of that tumour and obviously poorer survival. So very, very interesting stuff there. He also went on to show that in a treatment setting, so this is when he applied the vaccine I've been talking about. What you want to focus on here basically is the difference between this purple line and this, this orange-brown line here. So this is the effect of the vaccines. You can see these mice have preferential survival compared to untreated mice. When they've been exposed to isoprelin, again, the vaccine can't work as well as it did without stressing the mice, basically. So the mice succumb to disease much quicker. So it can impact on the natural development of the tumour, also potentially impact on our immunotherapies. And we think this is because triggering this stress pathway is really suppressing the response. So there's less of a response there that can respond to our vaccine. Obviously, a lot more work to do in this area. What cells are involved, etc., etc. But it'll keep Michael busy for the next couple of years. All right, in the last couple of minutes, I just want to point out some other work that's been done at the Diamantina Institute on lymphoma. There's only a couple of us really focusing on lymphoma as a disease, and you would have seen Mahir's picture pop up before in the video. So Mahir's the Leukaemia Foundation Chair in Blood Cancer Research, and he just happens to be located at the Diamantina Institute. He's basically leads our work there. He's a hematologist, and he's really looking at identifying better prognostic indicators in a certain type of lymphoma called diffuse large B-cell lymphoma. Okay, prognostic indicators meaning the ability to be able to predict prognosis or outcome in the patient. So the current methods for predicting this in non-Hodgkin's lymphoma traditionally is the IPI, International Prognostic Index, combined with cell of origin and also interim PET CT scanning. I hope I'm doing this justice, by the way. This is Mahir's work, and I'm trying to explain it for him. But basically what he's quoted is that although we have these methods, it still fails to identify many patients that will relapse with disease, and also it tells us nothing about how the malignant cells interact with the immune microenvironment. Okay, so he's also focusing on the immune system and trying to generate this, what he calls an immune score, okay, to try and stratify patients based on immune phenotype and their immune repertoire that's in this microenvironment. So what they use in their lab is a technology called nanostring. Basically, this is one method of looking at gene expression. Okay, so what they do is they've picked a range of genes that have been known to control cancer, so that they're the genes you want upregulated, and also genes which is termed immune checkpoints here, which are known to be bad. So upregulation of these genes, immune genes, you have less likely chance of the immune system controlling the cancer. So what he's done is try to stratify patients based on the ratio between these immune effectors over immune checkpoints. So if you have a high ratio, you have more of these effectors, they're the good players and less of the bad players. If you have a low ratio, you have less of these good players and more of the bad players. Okay. And when they divided patients based on that gene expression analysis, what they found very strikingly is these are the same patient cohort here looking at event-free survival and overall survival. What they found is if you look at those with a high immune score, okay, gene expression, they're the patients which have much higher survival levels compared to those with a low immune score. Okay, and you don't get this sort of spread in survival when you look at other traditional prognostic indicators. Okay, so again, quite early days, but there are definitely methods you can use which focus on looking at the immune system, which can stratify patients, and the likelihood that a patient will respond, will have a better prognosis, I should say, than others. All right, and the other work they're doing, which we actually collaborate with him on, is looking at immune suppressive myeloid cells. So I think I briefly mentioned earlier that there's immune suppressive cells which interfere with our immunotherapies and are generally bad for the immune response and can promote tumour growth. So looking particularly at these monocyte populations, and they've shown that in lymphoma patients, they tend to have much higher levels of these particular subset of monocytes in the blood compared to a healthy population. And if you specifically look on these monocytes, he's shown that in the cancer patients, again, these monocytes express high levels of arginase, which is an immune suppressive molecule. It's an enzyme which suppresses the immune response. Okay, so obviously if you have a lot of this stuff floating around, it's potentially bad case scenario. And if you spike these monocytes into a dish with T cells, so T cells you can induce to proliferate in a dish. If you spike in these monocytes from patients, you can actually prevent those T cells from proliferating as well. Okay, just showing that you put in these monocytes, you can actually inhibit the proliferation of the T cells, which obviously again we don't particularly want to inhibit in the patients. So we went and looked in the mice, the same lymphoma mice that we use, and we found that even in the mice there's a strong correlation here. This is a correlation plot between the levels of monocytes in the blood and tumour cells. Okay, so the more tumour cells you have, more monocytes you have, potentially indicating that the tumour cells could be pretty much driving this mobilisation of myeloid cells from the bone marrow into the blood, and that's promoting tumour growth. So Sarah in the group is focusing on this work now, and she's basically comparing monocyte populations from healthy mice or mice with lymphoma, and what she's observed is if you just focus on the monocyte population here, again this is flow cytometry, this allows us to detect different populations in the blood. What you see is there's a bias from a Ly6C high monocyte population to a Ly6C low non-classical monocyte population. So something about the cancer which is potentially driving conversion of one monocyte subset to another, or preferential survival enrichment of this population. And we think that this population could be the immune suppressive population, and Sarah's now doing assays to confirm that. It's basically showing that if you look in the bone marrow, again, this is where the tumours are in the bone, spleen in these mice. Over time, these Ly6C high subsets and Ly6C low subsets don't really change in the bone marrow, even though the tumour's increasing. But in the periphery, in the spleen, also I think we've seen this in the blood, we get this crossover. So there's a decrease in Ly6C high, an increase in decrease in the classical, increase in non-classical monocytes. So what consequence that has, we don't know yet, but we think it's related to that same observation that he is seeing in patient blood with these monocytes. And this is just showing that, again, if you look at correlation, if you focus on the Ly6C low population, there's arguably even a stronger correlation here when you look at comparing to tumour cells. So work going forward really is looking at are these monocytes suppressing the immune response against lymphoma, and what can we do then to eliminate or reprogram these monocytes? Because there are certain things you can do, again, by immunotherapies, etc., which can reprogram these cells to make them more able to help with the immune response rather than inhibit the immune response. Okay, this is my last slide. You're probably sick of me talking, so I'll stop. So where to from here with all this? So really, going forward, we're aiming to optimise our current vaccine strategies with the goal of really trialling these in clinical trials in non-Hodgkin's lymphoma soon. And to really now focus on looking at the lymphoma microenvironment and what immune landscape is in that environment. To firstly establish better treatments and treatment combinations. So if we understand more about the microenvironment, we can establish better treatments. But also to more accurately predict which treatments will be more effective in which patients. Okay, this is more idea about personalised medicine and tailoring treatments to certain patients. And with the stress stuff, we really want to provide substantial evidence, at least in our mouse models, of the negative impacts on stress, really to promote potential psychosocial interventions for cancer patients and for survivors that aim to sort of manage stress and promote well-being, if that's what's going to help. And like I said, please feel free to come and visit us or contact us. We're very happy to talk to people. We kind of get sort of trapped in our environment and ostracised from the rest of society sometimes, so it's very good to get an opportunity to talk to you guys and to show you around the lab. So feel free to contact me or the Diamantina Institute. That's my details and phone number there. It's been a pleasure talking to you, and I'm happy to take any questions. I'll try and answer any questions you guys may have. Thank you. I don't believe of any therapeutic vaccines that exist for lymphoma that's clinically available. Yep, that's what we're developing. The hope is really to combine therapeutic vaccination, which is aiming to boost that immune response, to combine that with conventional treatments. Certain types of stress are good. So some acute stress actually can boost the immune system. Okay, can enhance the immune system to a certain degree. We're looking at chronic stress over time. That sort of low level, that sort of stress that kind of eats away at you over time, low level, where you have all these sort of stress mediators getting pumped through the system at low levels for a long time. And that's been shown in plenty of different disease settings over time that can decrease immune function and increase potentially susceptibility to disease, or once you have disease, to worsen that disease. But everyone's different, and everyone's threshold is different, right? So a certain level of stress for one person, which may increase their risk to a certain disease that may not even exist in another person. So it's very complex. And that's where genetics comes into play. We can look at differences between people, basically, and look at their susceptibility to diseases and stuff. But it should enhance. So a lot of these oxidative radicals and oxidative species, etc., get released by these immune suppressive cells. Okay, these myeloid cells in the body. So potentially antioxidants, which can reduce a lot of those nasty players, could potentially help with immunotherapy because it will eliminate some of that immune suppression which is caused by those interactions. We've done those sort of studies in mice. So we have mice which are immunocompromised. They lack T cells and B cells, the main players, and these therapies basically don't work as well. So we have to be mindful of the fact that a lot of patients will be immunosuppressed. Okay, what level of immunosuppression will then determine whether you'll get any effect of these therapies is basically unknown. Okay, it depends on the type of therapy, it depends on the state of the immune system. So the goal really is for us to use the vaccines to re-establish or to help boost that immune response, to get it going again. And obviously it's not going to work very well if you're under high-dose chemotherapy because chemotherapy is going to keep knocking it down. Okay, so for example, if it was in combination with chemotherapy, potentially how we could use it is use low-dose chemotherapy, it's called metronomic chemotherapy, low dose over time, which is enough to kind of aggregate the system and get some tumour cell death, and that actually benefits promoting the immune response as well, but not enough to suppress the immune response. And then we come through with our immunotherapies to boost that response. Plus, there's going to be less toxicity associated with the chemotherapy because it's at much lower doses. So the goal really is not to replace conventional therapies with immunotherapy, but maybe to restructure the current therapies, or repurpose them, or give them differently. For example, lower dose chemotherapy to get rid of some of those side effects and nasty things, but allow it to work together with your natural immune system.